Welcome to the PubMed Daily Review for April 04SD. Today we scanned 2,690 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, sleep, pain, and neurological health. This trend explores the interconnectedness of sleep, pain, and neurological function, examining their impact on health and disease. This cluster covers 445 papers. Representative work includes evaluation of a contactless sleep monitoring device for sleep stage detection at home in a healthy population. Prospective study in free-living conditions, impaired dynein function preserves spinal interneuron survival and positioning in an ALS-like mouse model, and patterns and predictors of variability in patient-generated daily pain severity collected via a mobile health smartphone app. Trend 2 of 5, biomaterials and medical device design. This trend explores advanced materials and design for improved medical devices and treatments. This cluster covers 356 papers. Representative work includes evaluating chemical and surface stability of thermoformed and 3D printed clear aligners after intraoral exposure, practical research on the boundaries of MIDYA design principles with ceramic products as the carrier, and editorial note. An integrated method for the leakage fault mode diagnosis and life prediction of the reactor coolant pump. Trend 3 of 5, digital health and user experience. These papers explore how digital technologies impact health outcomes and user engagement across diverse populations and health conditions. This cluster covers 622 papers. Representative work includes development and launch of a Dutch mobile app, MediMama, on over-the-counter medication safety during pregnancy and breastfeeding, development and usability study, exploring nursing students' experiences in a brief virtual reality-enhanced workshop, cross-sectional exploratory study, and additive and multiplicative effects of socially stigmatized identities using linear regression to model effects on self-reported overall health as reported in the All of Us research program, quantitative analysis. Trend 4 of 5. Disease diagnosis and treatment strategies. These papers focus on improving disease diagnosis, classification, and treatment effectiveness across various medical conditions. This cluster covers 527 papers. Representative work includes preventive strategies against brain metastases, current state-of-the-art and future directions, reliability of the CPK classification assessed on long leg X-rays in patients undergoing total knee arthroplasty, and consensus definition of rapidly progressive interstitial lung disease, a critical unmet need to be led by pneumologists. Trend 5 of 5, genetics, evolution, and crop improvement. These papers explore how genetic and evolutionary principles drive adaptation and improvement across diverse organisms and traits. This cluster covers 740 papers. Representative work includes diversification of cichlids in the genus Danakillia in the Danakil Depression of Eritrea and Ethiopia, lifestyle-associated variation in type 4 secretion systems between phytopathogenic and environmental Ralstonia, and associations of tumor necrosis factor alpha genetic variants with metabolic syndrome and type 2 diabetes mellitus in a Thai population. Up next, our first paper out of 5, a double-negative prostate cancer subtype is vulnerable to SWI/SNF-Targeting degrader molecules by Philip Thienger and Mark Rubin from Department for Biomedical Research of Bern, Switzerland. Let's break down its key contributions. Prostate cancer isn't a single disease. It's a complex group of conditions with varying molecular characteristics. One particularly aggressive subtype, which doesn't respond to androgen receptor therapies, relies heavily on WNT signaling. This subtype represents about 10% of castration-resistant prostate cancers, making it a critical area for new therapeutic strategies. This study investigated a novel therapeutic approach for a specific prostate cancer subtype. Researchers utilized cell lines and animal models that mimic the double-negative, WNT-dependent prostate cancer. They tested the efficacy of SWI/SNF-Targeting degrader molecules, aiming to disrupt the key molecular pathways driving this aggressive cancer. The results were promising. Treatment with SWI/SNF-Targeting degrader molecules led to a significant reduction in tumor growth in models of the double-negative prostate cancer. This was achieved by interfering with the critical molecular axis that defines this specific cancer lineage. Importantly, this approach showed effectiveness against the androgen receptor negative, WNT, signaling-dependent subtype. This research highlights a significant vulnerability in a challenging prostate cancer subtype. SWI/SNF-targeting degrader molecules show great promise as a novel therapeutic strategy, particularly for the 10% of castration-resistant tumors that are androgen receptor negative and WNT dependent. While these findings are exciting, further clinical investigation is crucial to determine their real-world impact on patient outcomes. Up next, our second paper out of 5. CD3 Nanobody-Engineered Extracellular Vesicles Driving in Vivo Generation of TCE-secreting CRTS for Solid Tumor Therapy with Memory Response and Minimal Immunogenicity by Shiwei Huang and Drian Cho from Translational Cell Therapy Center, Department of Medical Research, China Medical University Hospital, Taichung, Taiwan. Let's break down its key contributions. Chimeric antigen receptor T-cell, or CAR-T, therapy has revolutionized cancer treatment. However, generating these powerful cells often involves complex lab procedures and potentially risky viral vectors. Researchers are exploring ways to create CAR-T cells directly inside the body, or in vivo, to simplify treatment and improve safety. This study investigates a novel approach using natural nanoparticles called extracellular vesicles to achieve this goal. The researchers engineered tiny, natural carriers called extracellular vesicles, or EVs. They modified these EVs to specifically target CD3 plus T cells, the very cells we want to turn into CAR-T cells. These engineered EVs were then loaded with the genetic instructions for a special type of CAR-T cell, one that can secrete a bispecific T-cell engager. The study then tested if these EVs could successfully deliver these instructions to T-cells within a living organism. The results were highly promising. These engineered EVs demonstrated significant anti-tumor activity across different solid tumors. Importantly, they showed minimal signs of triggering an adverse immune response, a key advantage over existing methods. Furthermore, the treatment led to a greater proportion of CAR-T cells with memory capabilities, suggesting a more durable and long-lasting anti-cancer effect. This study presents a significant advancement in CAR-T therapy, showcasing extracellular vesicles as a promising, natural delivery system for in vivo CAR-T cell generation. The reduced immunogenicity and enhanced memory response observed are particularly exciting. While these findings are compelling, further studies will be crucial to refine this technology and pave the way for its potential clinical application in treating solid tumors. Up next, our third paper out of 5, Unifying the Odyssey, Artificial Intelligence for Rare Disease Diagnosis and Therapy by Mylon Ho and Richard Sidlow from Department of Radiology, University of Missouri, 1 Hospital Drive, Columbia, Missouri, 65212, USA. Let's break down its key contributions. Millions of people worldwide live with rare diseases, facing a long and frustrating journey to diagnosis, often taking 5 to 8 years and involving multiple specialists. Despite advances, less than 5% of rare diseases have approved therapies, and the economic burden is immense. This review explores how artificial intelligence can revolutionize this landscape. This paper is a narrative review, drawing on multidisciplinary expert insights. It examines the current hurdles in rare disease diagnosis and therapy, highlights recent breakthroughs in AI, and proposes a future model where AI proactively integrates diagnosis and treatment, aiming to transform patient care from reactive to proactive. The current reality for rare disease patients is a lengthy diagnostic odyssey, averaging 5 to 8 years, with less than 5% of diseases having approved treatments. AI promises to fundamentally restructure this, collapsing the traditional sequential pipeline into a single, integrated engine. This creates a powerful feedback loop where precise diagnosis directly informs and accelerates the creation of bespoke therapies, potentially at scale. The integration of AI holds immense promise for rare disease care, potentially transforming it into a proactive, closed-loop system. However, significant non-algorithmic barriers remain, including biological modeling and manufacturing challenges. The path forward necessitates a systematic alignment of computational, biological, and economic infrastructure, guided by expert human feedback to truly deliver personalized cures at scale. Up next, our fourth paper out of 5, Sleep Disturbance Trajectories During Childhood and Early Adolescence Associated with Increased Suicide Risk by Rebecca S. Huber and Bonnie J. Nagel from Oregon Health and Science University, Portland, Oregon. Let's break down its key contributions. We know that sleep problems can be a sign of distress, and they've been linked to suicide risk. But what happens to this link as children grow into teenagers? This study dives into how different patterns of sleep disturbance over time might affect a young person's risk for suicidal thoughts and behaviors. Researchers looked at data from over 11,000 children in the Adolescent Brain Cognitive Development Study. They used a method called latent class growth analysis to identify different patterns of sleep disturbance over 4 years. These patterns were then compared to rates of suicidal thoughts and behaviors, using a standardized assessment tool. The study identified 3 main sleep patterns. Those with a high decreasing pattern, meaning sleep problems started high but improved, had a 1.75 times higher odds of more severe suicidal thoughts and behaviors. Even more striking, those with a moderate increasing pattern, where sleep problems were moderate and worsened over time, had a 2.34 times higher odds. These associations were statistically significant. This research highlights that it's not just about having sleep problems, but the pattern of those problems over time that matters for suicide risk. Both improving but initially high sleep issues and worsening moderate sleep issues are red flags. This underscores the importance of paying attention to sleep in young people as a potential target for suicide prevention efforts. Up next, our 5th paper out of 5, Vesicles, a human-evolved serine integrase system for efficient large cargo genome integration by Alfonso P. Ferruggio and Ruby Tsai. Let's break down its key contributions. Many debilitating genetic diseases stem from mutations within large genes. The challenge in gene therapy has been efficiently delivering and integrating the full-length, functional gene to correct these defects. This paper introduces a novel system designed to overcome this hurdle, paving the way for broader therapeutic applications. To address the need for large fragment gene delivery, the researchers developed Extracellular, a novel serine integrase system. This system was engineered and tested in human cells, both in stable lines and through transient transfection. The key innovation lies in its ability to recognize a specific endogenous site in the human genome for precise integration of large DNA payloads. The results were highly encouraging. In stable cell lines, the Extracellular system achieved integration frequencies of up to 32% for a 10 kb plasmid. Even with transient delivery via transfection, they observed impressive knock-in rates of up to 13%. This represents the first serine integrase system evolved in human cells to target a native genomic site. This study presents a significant advancement in gene editing technology. The Extracellular system's ability to efficiently integrate large DNA fragments opens new avenues for treating genetic disorders previously considered intractable. While promising, further studies are essential to translate these findings into clinical applications, focusing on in vivo performance and safety.